record. Okay. So, first of all, we will read this protein. We have to understand this protein. So, you can open these files with Notepad. Okay. Or Notepad or WordPad. So, I will open with, now at present, I will open with WordPad because it could be highlighted. So, it is the program written. With this only, the any molecular docking simulation softwares will open the build the protein. Okay. So in this, if you see this, it is a oxidoreductase protein. Okay. So basically, it is considered with oxidation and reduction. So it is the crystal structure of human aldose reductase. So the protein name is human aldose reductase, and there is one inhibitor which is present in this, and the inhibitor is zen-aristat. So you can see there is an inhibitor which is zen-aristat, and it is having only one chain, that is chain A. So you will get lot of information in this, and it is related to the organism Homo sapiens. And then it is related, it is expressed in a system which is a bacterial system, that is E. coli. So it is an oxidoreductase enzyme, human aldose reductase with an inhibitor, chain A is present. It is for from Homo sapiens, expressed in E. coli. When you scroll down, you can see the resolution. It is 2.5 Armstrongs. So when you take any protein, so you can go for lesser than 2 if the resolution will be better. So you can take 2.5 also, but if it is less than 2, it is better. So then if you give control F and give missing, so if you give missing, it will show there are some missing residues in it. So it is the first amino acid, which is methionine, is missing. So we get some information related to this. So it is methionine which is missing. Suppose if it is at the active site, we have to build this. Otherwise, there is no need. You can build this with the Swiss PDB viewer or Chimera. When you scroll down, so you can see there are several remarks, and if you scroll further, it will show the atom-wise distribution of this protein. What are all the atoms present in this protein? So it is having number of amino acids is 316, but if you count the atoms are numerous. So one is nitrogen, the second is carbon acceptor, next is carbon, next is oxygen. You can see this. So if you scroll down, you can see the different atoms and their XYZ coordinates. So these are the XYZ coordinates. Suppose if you scroll down further, then if you come down, so here you can see this is the terminal end. So this is the end of this protein. So totally there are 315 amino acids. So it is the end of this protein, which is phenylalanine. Then you can see here there is het atom. So het atom is present. So we can give one more color. So it is het atom. So these are heteroatoms, the atoms which are not related to the protein. Okay, here you can see different atoms, which is from N-nicotinamide adenine phosphate. That is why phosphorus is coming. So if you scroll down further, so you can see there is one more heteroatom, which one molecule is present. That is zen-aristat. So here that is zen-aristat. So first heteroatom is NAP or NADP. The next is the zen-aristat. Then if you scroll down further, so there is another het atom which is water. So water is also not belonging to the protein. So it is also called as heteroatom. But one more point to be noted is that there is no hydrogen which is present in this because it is molecular size is very less. In crystallography, it is very tough to represent this. That is why we add hydrogen in the docking simulation programs. Then if you scroll down further, you can see the connect records, how these atoms are connected. That is the records of this, connecting records of these atoms. So you will get lot of information from this, what type of enzyme it is, whether it is having, how many chains are present, whether an inhibitor is present, whether it is expressed, whether it is having mutation or not. It is through the submitted through the X-ray diffraction. Then you can see the missing residues. So here I have already represented there is one missing residue, that is methionine. Then if you scroll down, the atom distribution, their XYZ coordinates. Then if you scroll down further, you can see the terminal end of this protein, which is phenylalanine. Then the het atoms will be there. So here it is NADP. Then it is followed by Zen-aristat, and it is followed by water. Okay. So one is Zen-aristat and the next is water. So then there will be connect records. Okay. If you open in the WordPad, I can give the color options, but in Notepad, it is there is no color options. Okay. So I will close this. I will give don't save. Okay. So now we have understood this protein. Okay. So for before going to any molecular docking simulations, you have to understand the protein first.